We did 4 synthetic biology projects: X-verter, predator-prey, human encryption, cancer Stickybots, and bacterial dynamo. So Nicholas Tang, Austin Hines, Ling-Chong Yu, and myself have over the summer been trying to design 2 unique synthetic oscillating gene circuits that operate at the population level. And we've also explored computational chemistry to study how quorum sensing systems work. So for computational chemistry, what we did is we looked at these 4 AHL-based small molecules for the Lux, Syn, LAS, and RHL small molecule systems. And we looked at the HOMO-LUMO gap. And we saw that these numbers that you see for the gap figure are very similar. And after looking at the common common oxidation points on the HSL backbone, we were able to fit a curve to Dr. Ling-Chong Yu's experimental data on Lux pH degradation. And we extended this to the other small molecule systems. This enabled us to do a lot more extensive modeling. So the first circuit we designed was the X-verter. This is a 3-stage oscillator that works on the principle of forward activation and back repression. And so you can see it activating or oscillating between the 3 different fluorescent proteins that will be expressed. So this is the actual circuit that we designed. And then to model it, we derived a system of differential equations. And then we actually modeled it. And so you can see in this model that we have stable oscillations. And all 3 different fluorescent proteins are activated at different stages in an oscillating cycle. The second circuit that we built was a predator-prey ecosystem. And imagine that the prey is a source of food for the predator cells. As the predators eat the prey, the population of prey decreases. And as there's less food, the predator population decreases as well, so that the prey population rises again. And this gives rise to oscillations. So then to model this, we derived another set of differential equations. And then we have our actual model. And you can see that we got oscillations with the predators slightly following the prey in oscillations. To counter the complexities of doing 3A assembly on a massive scale, we developed some software, an IDE. And what this software does is it actually lets you automate the entire assembly process on paper. So you can actually just look down and know exactly what you need to transform where, what you need to ligate to what to get the entire process to work. It also lets you check sequences to see if what you transformed is actually what you're supposed to get. And one of my favorite features is that it actually down-syncs with the standard registry of biological parts. So you're always up to date. So in conclusion for this project, we derived pH degradation rates using computational chemistry. We designed and modeled the X-verter, a 3-stage synchronized oscillator. We designed and modeled predator-prey, a 2-stage synchronized oscillator. And also, we're almost finished with assembly for this circuit. We developed BioBricks Manager, a biological circuits IDE. And it's going to be released as open source soon. We also performed experimental characterization and obtained specificity and latency graphs for characterization.
All right, my name is Pat O'Brien, and I'm the designer of the Human Encryption program from Duke University, which is basically a system for encoding information in vivo with DNA and light. I worked this summer with Keddy Chandran, Austin Hines, and Dr. Fan Yuan of Duke University. So the basic idea of human encryption is that you can take a bacterial symbiote that you've programmed with a luminescent operon, and you can insert this in vivo and have this actually colonize in the cecum of your host, and you'll be able to detect luminescence using CCD cameras, which will be a signal of presence of a message that's either coupled with the luminescence or will serve as a message itself. So the first step we did is for coupling with a message, we wanted to make codons serve as an encryption scheme for information so that you could actually use DNA as a message system itself. And the second phase of what we had to do was actually develop this library of different wavelengths of light so that our light could serve as messaging systems and signify different sort of things. So we had to engage in a series of site-directed mutagenesis experiments in order to create our library, starting with a blue-green operon, and we engaged in as much redshifting as was possible. And so you can see here the results of some of our experimentation, how we've actually moved our peak more towards the 500 nanometer range. And here is the data from all of our independent mutation experiments and our combination mutation experiments showing that we have actually made this operon into a library of wavelengths, and we intend to expand this more with random mutagenesis. And so to actually test that our system was viable, we used germ-free mice and constructed an E. coli system carrying this Lux operon, which was optimized to operate at 37 centigrade, so it will work in a mammalian system. And it constitutively expresses its own substrate, so it will constitutively glow in system. So what we did is we orally dosed our mice, and as you can see, the picture on the left is actually at the time of administration. We did imaging, and you can see that our bacteria are still localized in the mouth and in the stomach. But 17 hours later, this image on the right, you can see that as we expected, the bacteria localized to the colon itself, and especially the cecum, where you can see them colonizing and growing. So you may be wondering what some of the applications of this technology might be. Well, if you're carrying around a glowing bug that's in your gut, it's a good way to signal that something there is in presence that shouldn't be there. So what you could do is you could use this as a marker system if you can get the bug into infrared range. So you can have infrared light coming out of somebody's cecum, and you can detect that very easily with relatively simple technology, CCD cameras. And what you can do is you could couple that with a message encrypted in DNA. And what you could have for the security aspect is when you detect this luminescence coming out of people, you can pull them aside, and it's a simple matter of recovering a stool sample and running PCR to actually get the message back out. I know it doesn't sound pretty, but it could be fairly effective, and depending on how you want to actually apply this, it's relatively undetectable to the host. But a more interesting and more perhaps useful application is if you develop a sophisticated enough library of wavelengths of this operon, what you would be able to do is combine light with hormone sensors or biosignaling sensors so that you could have a sort of visible health thermometer, so that when some sort of hormonal imbalance comes or some sort of bacterial signaling comes in, you can start glowing a certain wavelength of light, which you can then look up on a standardized table and say, oh, 498 nanometers means this is colonizing or this is wrong with this person. So you could have sort of a visible way to diagnose somebody in a very simple manner. And so at the end of the summer, what we actually did manage to do is we constructed our bug. We put it in our mammalian host and got our imaging done. And the next phase is to incorporate our operon into the chromosome so that we don't have to worry about antibiotic injections to maintain plasmid. And then the next phase after that is simple message insertion and recovery. So overall, mission accomplished on a very simple proof of principle kind of experiment.
Thanks, Pat. All right, so here is the Cancer Stickybot project. It's a project me and Ling-Chong Yu came up with together, and also Nirav Lakhani worked on. Here I'm going to basically be talking about what we actually did, which is develop a novel suicide circuit for tumor-targeting bacteria. So a lot of you guys are familiar in pop culture with the book Prey, which is an example of micromachines and swarm behavior. Basically nanobots that synchronize their behavior to act as a group. And we're using these properties found naturally in bacteria to engineer a system that will allow us to use bacteria in a way that kills cancer. So here are actually some colon cancer cells. The idea is basically that we have our bacteria stick to them, and then after they stick to them, they deliver a toxin to them, and that kills them. And then after they've killed the bacteria, they actually commit suicide. So this is the first component, targeted localization, making a sticky swarm that actually stick to this cancer cells. And how do we do that? I'm just going to break down this circuit really quickly. We're using nanobodies, a nanobody display system. It's actually, research is currently going on in Madrid, Spain, but we're expressing a very small single domain antibody on the surface of bacteria, in this case anti-carcinogenic embryonic antigen, which is very common in colon cancer. And this allows our cells to actually target the tumor. Here's a picture. And then so for discriminate killing, we have to find a way, once they get there, once they stick, how are they going to kill them? Well, they're going to kill them by not only invading them, but also producing a small molecule known as 5-FU or 5-fluorocytosine. And that's what actually is the toxic agent that kills these things. So quickly going through the circuit, you see invacin and cytosine deaminase linked to quorum sensing. So once they're in that environment, they turn on and it's kill time. So the last circuit, this is a circuit that's actually built, and we're going to tell you about the experimental results for. This is, so when you introduce bacteria in a system, if they don't go to the right place, what do you want? Well, you want them to kill themselves. You want them to also kill themselves once they've done their mission. And that's what we've programmed into these guys. Stickybots self-destruction. Okay, I'll go through the circuit right quick. We're just linking quorum sensing again. Actually, in this case, to CCDA, which is an antitoxin. When these guys are together, they're protected. They're all producing an antitoxin. They're in the safe zone, all right? But if they become isolated, if they drift away from the tumor environment, or they kill their target and then they, dissociate and go, outside of the concentrated area, they commit suicide by CCDB, which is a toxin that poisons the DNA gyrase. So here's a picture of CCDB interacting with gyrase A59. It inhibits it, and DNA gyrase is what untangles DNA. If you don't have that working, you die. Okay? CCDA is what inhibits CCDB. So, like I said, these guys are in the quorum sensing environment of the tumor. They survive. They produce CCDA, cancels out CCDB. They're isolated. They're constitutively expressed CCDB. They die. So here's the experimental results, experimental data that we were able to collect. This graph puts it a little bit more clearly. So we have a high cell density, followed by a starting low cell density. As you can see, the ones at the high cell density, they grow up to a peak around 1 of OD600, whereas these at the low cell density, they fail to grow up, right? But in this last case, we said, okay, well, not only do we want to see that they don't grow up at a low cell density, we want to know why that is. And the reason why is because they depend on that AHL to activate that antitoxin. And you don't have sufficient AHL, you get death. So here we're actually not only recovering the original high cell density population, we're actually going beyond it with a very high concentration of AHL. Here is the experimental the modeling that we've done on this for the whole entire system. Mission accomplished for this part, but we still have the first two parts to do, so remember that.
Hi everyone, Merrick Josephs. This is my associate Hattie Chung, and our advisors Tom Levine and Jingdong Tan. And I worked on the bacterial dynamo project. And the question you're probably asking right now is, what's a dynamo? Well, a dynamo is it's a generator of electric power by a spinning magnetic field within a coil. So by Faraday's law, it will generate electric power. But we wanted to do this with bacteria, so we used our good friend Magnetospirillum species A and B1, which is a species with one flagella on each side, but they grow magnetic crystals in the middle. So what we wanted to do is basically get these these flagella to stick to a surface above a coil. So once the surface is, once this flagella is stuck, the motor's still running, so the cell body is going to spin. Once the cell body with magnetic crystals are spinning and it's over a coil, it should generate electric power. And the way you do this is that flagella are sort of easy to modify because they come with this variable region in the middle where you can sort of express stuff on the outside. So what we did was, oh, well, here's actually the bacteria spinning around the coil generating power. I like that picture. So what we did was we screened for the short peptide sequences that would just naturally bind to this hard-baked photoresist, which is this material you could pattern really small and accurately, and so we figured that'd be a good size for bacterial colony to go onto this, this coil. Where we got stuck is we had to kind of cut out this AMB1 gene, which is the magnetic bacteria flagellin gene. We kind of got stuck with that. So did Brown team, though, so I mean, we're not alone on this. And so we make this fusion protein by taking this sticky peptide, put it in the, so it gets expressed on the outside of the flagella, put it back into the magnetic bacteria. What we also have to do is make a coil. I'm an electrical engineer, so this stuff's interesting to me, but you're all biologists, so if you really want more information, I could talk to you about it later. My poster's right out there. But this is what it looks like. I'm very proud of it. And it's about 0.5 centimeter by 0.5 centimeter. And So our coil is complete, and we isolated the sticky peptide, but we're currently working on making this fusion AMB1, this protein that will make it stick. And some of the applications I always thought would be interesting is that people are doing research to try to get bacteria to sort of convert chemical energy of like pollutants or nuclear waste into bacteria energy. And if you could do that, then you could make a battery out of anything. Or not a battery, a dynamo out of everything. So yeah, here's our conclusions.
All right, so very quickly in conclusion, us as a team, we've characterized a system for small molecule communication, in particular our 3-stage X-verter and our 2-stage predator-prey. Also, in that process, we've created an open source gene circuit interactive development program, which is basically a biobrick management tool, if you will. Also, for human encryption, we've created an alphabet for communication via a molecule medium. We've done this through our codon-based encryption scheme and our light library. Also, for cancer Stickybots, we've developed a working bacterial circuit to basically cause bacteria to self-destruct outside of a tumor environment. Also, lastly, we've evolved E. coli to stick to a photoresist and engineered an apparatus for bacteria, magnetic bacteria, to spin above a coil, inducing electric current in that coil, also known as our bacterial dynamo. So, we'd like to finally acknowledge our faculty advisors, Jingdong Qian, Fan Yuan, Tom Labine, Lingchang Yu, Faisal Reza, Myra Halpin, and Bob Gottwals, and also our collaborators from around the world listed on the right. And also, we'd like to give many, many thanks to our investors, the Lord Foundation, the Pratt School of Engineering, and the Institute for Genome Sciences and Policy. Ladies and gentlemen, thank you very much.
This system's actually been built before, but they didn't use sticky flagella. They actually used antibodies. So I mean, so it degraded fast. But in their system, which was made kind of crumbly because it was sort of they used a little coil and it's just a bunch of bacteria, and it was like a high school science fair. But they got about 10 microwatts, wasn't it?
It was microwatts on a 1 centimeter by 1 centimeter square load.
Which I mean isn't bad, but it's also unoptimized. And I mean, flagella are pretty efficient.
So for you non-electrical engineers, that's what, like a AA battery?
No.
No, I mean for the whole system. For the whole system.
Well, I mean, if you make it big, then yeah. I mean, but
I think he actually out of his system, his whole system was producing about a AA battery.
It was about 1.5 volts or something.
But it's basically it's promising. Is basically what it is. That's a decent amount of energy for a half a centimeter by half a centimeter thing.
Right.
You said for the cancer-killing bacteria that they made 5-fluorouracils?
Right, that's what we're actually programming. Cytosine deaminase is an enzyme that converts 5-fluorouracil, 5-fluorocytosine, 5-fluorouracil. Right.
I see. So you would give a person 5-fluorocytosine and then the bacteria would convert it to 5-FU?
Right.
Okay.
Exactly.
Now for the cancer Stickybots, as I understand it, the tumor, because of the adhesion, creates a high cell density, which inhibits, I guess, synthetic apoptosis in this case. How would you maintain of these bacterial cells when initially introducing them to a biological system?
Exactly. Yeah, that's the main question. There's an immune system, right? So I mean, we pretty much have to say knock it out with like rapamycin and cyclosporin, something like that.
In the first part of your talk, what was the experimental characterization you mentioned?
Right, yes. We actually collected that on Thursday, and we have the graphs if you want to see them, but we're still doing an analysis on those. We actually characterized 3 circuits made at MIT, as well as a circuit we made in our lab. Those 3 circuits were the SYN circuit, last small molecule circuit, as well as the RHL small molecule circuit. And those definitely need to be characterized, and for some reason they haven't been characterized as yet. But we went out and got the small molecules, we have the circuits, and we have the actual results on whose laptop we're already making somewhere over here.
Yeah. So this is really like when we finish our data analysis, this is promising data that not necessarily completely helps our project, but it's going to help a lot of other projects for future iGEM competitions.
And second question, on the regulated suicide,
Right.
what was the actual device or two devices?
The mechanism, right. So it's a regulated suicide device. It's actually drawn out on the parts page, but that's not, we actually didn't build it. We're in the process of building it in the form of BioBricks, but the actual circuit we built was not in BioBrick format. We used circuits already present from Wing Chun Yu he had a constitutive expression of the Lux receiver as well as the Cinder device, and we coupled that with CCD, CCDB plasmid that allowed for the regulated suicide that you saw in those graphs.
Okay.
For generating your libraries, what type of random mutagenesis did you use?
We actually, for the course of the summer, did a bunch of site-directed mutagenesis based on one of the papers that you see on our citations on our poster. But we followed a very, very similar Lux operon mutation scheme, and we're just doing some site-directed things to see how much redshifting we could get out of their protocols. So the next stage is actually the random mutagenesis, in which we'll start playing around with the whole sort of operon and see what we can get out of it.
All right, last question, we have to move on. Well, this is about the bacterial encryption. How stable is the colonization in sequence? Does it last for weeks? Or?
That's one of the things we're still in the process of characterizing. It was very difficult to actually get the approval to begin putting this bug into mice, so we have only had about a week of time to play around with them, and there's been some hiccups in the process because it was sort of a different thing for people, the resources we've been using, so we still have to characterize that some more to be sure. And the other thing that we are aware of is, since we're using a germ-free system right now, we know that whatever data we get for that is going to be completely different when we actually translate it to a normal mammal system. So that's a lot more characterization.
Right. We did cheat. When you put them in, they're actually a small population, and then they grow up while they're inside the gut. And we're kind of cheating, though, because we're using antibiotic to prevent plasmid loss. Also, we're using, supposedly, mice that have a low germ content in the gut. In the future, we'll actually put it incorporate into the chromosome. It's done plenty of times before, and we'll be able to maintain long-term growing without the addition of antibiotic, without anything. The operon has everything necessary for life production.
Let's thank our speakers again.
Thank you.